Welcome to Faculty Focus, the podcast of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences of the University of Antwerp, live from our annual Faculty Research Day, where we highlight the research of our professors and PhD researchers. I am Teun.
And I am Marleen.
And just like that, we welcome our last guest of this year's podcast marathon. Welcome, Sarah.
Hi.
Sarah de Beuckeleer is a PhD researcher in Biomedical Sciences at the Laboratory of Cell Biology and Histology. She researches the drivers of cell state plasticity in glioblastoma with image-based contextual cell profiling. Glioblastoma, or brain cancer, why is there still no cure?
Mainly because the main reason that people still relapse is that there is like a small subset of cancer cells called GSCs, and they're very difficult to target. There's this cellular plasticity that causes the cells to constantly switch phenotype, and this is what makes them so hard to target.
Okay, and why is this type of cancer so aggressive?
Because it grows very fast, and also those GSC cells, they tend to invade strongly into the brain parenchyma, making it very difficult to resect.
Okay.
And you focus on cell state plasticity. Can you tell us more about this?
These GSCs, they have been defined using transcriptomic studies to contain four main cell states. So we have astrocyte-like cells, mesenchymal-like cells, oligodendrocyte-like cells, and neural progenitor-like cells. And those four main states, they have specific properties. They can also interplay with the brain parenchyma, the resident cells of the brain itself. And they invite strongly, which makes them very hard to target.
So to find these cells, you use a technique called cell painting.
Yes.
Tell us more about that.
So cell painting is a very easy technique. It's also developed to make our lives easier. Basically, it's leveraging one specific aspect of cells or cell identity that is often overlooked. Because when we look at those cells, we can also see that a neuron looks differently from an astrocyte, for example. Yet if we want to identify neurons and astrocytes in co-culture, we're still sticking to the methods that we know, using immunocytochemistry, for example, targeting specific proteins. But we can also use this morphology and use this to our advantage. And the cell painting approach stains not specific antibodies, not specific epitopes, but stains the morphology. And then we can combine information from the morphology with information from immunocytochemistry to train artificial intelligence, convolutional neural networks that are capable of predicting certain patterns and linking those morphological patterns to the cell state. And thus, in the future, making it possible to just look at the cell morphology and then output automatically what the cell phenotype or cell identity actually is.
And what do you use to give a color to the morphology?
The classical cell painting approach is based on some very simple dyes. So there's Histology just to stain the nucleus, some concanavalin, which is an ER marker, wheat germ agglutinin staining lectins, also the cell membrane, the Golgi apparatus, for example, mitotracker staining mitochondria, and the actin cytoskeleton is also marked. So these are just really generic structures that are present within every cell.
Okay. We can all be moved by a beautiful painting. When will you feel satisfied with the picture of your glioblastoma cells?
When I think a picture is beautiful, personally, I always like to look at the nucleus. It's also something that is, a lot of people within our lab are really enjoying to look at a beautiful nucleus. The actin, so the actin cytoskeleton often really gives beautiful images, because it's like this entire network within one cell. It's really beautiful to look at.
So those are the two things you focus on, or you see?
No, this is just the looks.
When you look at it, you say, a nice nucleus, a nice...
that's nice, that I would hang in my living room.
Well, why not, right? I think there's something marketable about this.
Yeah.
You also developed a method to position and visualize assembloids in flow as an alternative to traditional in toto imaging. What exactly are assembloids?
Well, assembloid is derived from the word assembly and also organoid. So if we're looking at thinking about organoids, then these are iPSC-derived models. So we start from a stem cell, we develop them towards NPCs in 3D culture, and then this 3D culture, we develop it further in vitro to resemble the human brain. So this is like a small brain in a dish, if you put it simple. And then we assemble it with something else. In my case, for my assembloids, or to study the GSC invasion, it's specifically with a small tumor. So there I take the patient-derived GSCs, make them into small three-dimensional structures, small spheres, so the tumors. And by putting those together, they start to fuse. They do this automatically without me having to do anything, basically, because it's the nature of those GSCs to invade into this brain-like structure.
Okay, and how do you image them? Or how do you get a picture?
There's multiple ways to study these three-dimensional structures. So they're approximately 1.5 millimeters in size. So they're somewhat too big for the light to get through. If we're just looking at it by eye, it's just a white blob. So one option would be to cut them to make slices of it, so that they become smaller. We can easily stain them and use regular microscopy techniques to visualize or what's inside of our sample. There's one difficulty, certainly for these assembloids structures, where we're looking at invasion of these GSC cells inside the structure. We don't know where they are, and I do really want to gain a complete view of where exactly, so the spatial location of these GSCs in my sample is really important. So therefore, I do not want to cut the sample, because then I lose a lot of spatial information. So to make it easier, or to retain this three-dimensional nature of my samples, I'm making use of tissue clearing technique, where I make the sample transparent so I can completely see through it, and the light can also, the light that's used by the microscope, can pass through the sample and also reach the detector. So the sample becomes transparent, and then I also use light sheet microscopy, which is a more light-efficient way to visualize these millimeter-sized structures.
I see an enlightened brain.
And you describe in your abstract, you describe the classical approach of imaging as tedious. Why? Can you talk about a little bit why you use the adjective tedious?
I believe in that case I'm referring to the cyclic immunofluorescence approach, because there's a lot of the immunostaining relies on antibodies, and antibodies do not always work. They depend on the sample, depend on the sample preparation, different conditions, also the supplier, differences from batch to batch. So this is something that's not always foolproof. It's also quite expensive, and I can only multiplex a certain number of channels. So I only have actually 3 slots available to combine different markers. But I have 4 subtypes, so I at least have to do 1 cycle of iterative staining to get a complete overview of what is exactly in my culture for those 4 subtypes. So that's why it's a bit tedious. I have to do a lot of work. Okay.
It sounds like a good word for the process. Yeah. So I read in your presentation, or I saw your presentation up front, and I saw that you want to combine 2 approaches to gain better understanding of patient-specific differences. Can you tell us a bit more about these approaches and why you think combining them will be a good thing?
During my PhD, I developed those 2 approaches, the ones that we already talked about, so cell painting and also the high-throughput light sheet microscopy approach. So the first, I want to apply to have a better patient stratification. So I talked about the heterogeneity within the GSC cell cultures. And this heterogeneity and also the plasticity, so cells being able to switch from one state to the other, is said to be linked to the invasion potential and the aggressivity, or the aggressiveness of your GSC culture. So being able to get a better understanding for one specific patient, if a patient is coming from the hospital, and this patient has a certain pattern or certain presence of specific subtypes, we can get a very easy and broad view of which subtypes are already present within this patient using this cell painting approach. So this on the one hand, and then on the other hand, we have this high throughput assessment of cell invasion. So the first approach was cell painting, so it's static, but it's just the different subtypes that are present within the patient. But we also want to see how these GSC cells are behaving or how they are invading within the human brain. So then we get also a behavioral readout. And I believe by combining both, we can really associate the subtype presence with how the cells are invading, and in that way get a better understanding of how certain treatments might work for the patient, but also maybe give a better idea on the prognosis, see how this patient is going to evolve, which drugs might eventually work, and also in further drug development pipelines, include all this patient variability to find novel drugs that can work on different types of patients.
Yeah, but the first application would also be individualized medicine, maybe.
Yeah, exactly.
We'll wrap up with one last question. What advice would you give a starting PhD researcher?
Fuck around and find out.
Okay.
A last bumper sticker.
That's a good one.
Of the day.
Okay, great. Thank you very much, Sarah, for coming by.
Yes.
And good luck with your PhD.
Thank you for the invitation.
Thank you, Sarah.